All right. So, please welcome Angie from the Fred Hutch, is it Fred Hutch Institute? Fred Hutch Cancer Center.
Yes.
Um, and I'll let you introduce yourself.
Awesome. All right. Well, thank you everyone for having me. It's super exciting to meet everyone. And for the introduction, Tim. As you said, my name is Angie. I am a post-bac research technician in NASA's lab, and I'm currently working in the Public Health Science Division at the Fred Hutchinson Cancer Center. And today I'm going to be talking about automation in academia and our lab's road into the early stages of integration of robotic automation in a mixed lab environment. So our lab is both computational and wet lab based. We have a space for both types of experimentation. But our question now, or like what we're trying to do, is we want to evolve our day-to-day bench work by using these automated tools and robots into integrating them into the wet lab space and trying to get the pace of integrating it to catch up to the efforts that we're making. But some of the barriers that we've had early on, and many labs have, is costs. These robots are not cheap, as well as the equipment and consumables that are needed for these robots can be very specific, and that cost can add up a lot. The Some tools are very purpose specific, so not all automation tools and robots are universal and can be used for many different resources. Those are they're barely being developed or they are slowly being integrated. Robotic maintenance, that's a whole skill that needs to be taught. It's basically engineering that has to be taught to biologists. And computational training, that's also another skill that is required to be able to like effectively get these automated tools integrated. And space. Sometimes some lab spaces, especially for us, like we struggled with organizing our space, considering that we had limited space to work in. So integrating these tools um, became an effort when it came to adjusting for that. But despite the challenges that these labs we face, or early starting lab or beginner labs face, there's an increased interest. Many of the benefits are like worth it in the long run. There's increased precision for protocols. There's reproducibility and protocol standardization. So you're able to get the same results every time that you run a specific protocol. Or there's laboratory efficiency, not only in like the time spent in lab, but also letting scientists be able to answer these or ask these unforeseen questions that otherwise would have never been able to been asked, given that they would have spent that time setting up for a experiment instead. And then there's the time saving and then scalability. So this graph is meant to show the growth in PubMed, the amount of publications that include the words automation or robotic. So like automation and automated is exponentially higher than robotic and robots, but that specifically has to do with like automation is used in a way to like describe data processing, and robots and robotic is more specific. But we can see that they're starting to catch up to one another. So people are interested in integrating robots into their wet lab experiments as well. So what we want to do is we want to close that gap between automation and robotics. Our ultimate goal is to integrate data generation and analysis into the same pipeline and to overcome challenges that are met with long-term experimentation methods. And the two main challenges that we're facing in our wet lab space is working with non-standard model organisms. So our model organism is Borrelia burgdorferi, which is the main bacteria that causes Lyme disease. The main barriers with working with this particular model is its safety level. It's considered BSL-2, and you have to go through specific training, and it has to be done in secure under like a biosafety cabinet in order to grow this particular culture. And that takes time to even train up to do, and then like to feel confident in being able to do these types of experiments. They grow in microaerophilic environments. Contamination is a big concern, especially if we're working in a shared lab space. Having to cross-communicate with other labs, these experiments that you're running, as well as what they're doing, and trying your best to keep your samples stable and sterile. The Borrelia itself has a particularly slow growth rate, so these experiments can run for a very long time, and they they need constant tending to. And in particular, Borrelia has like this tendency to lose its own plasmid if you aren't tending to it throughout its growth stages. Another challenge that we have is upscaling these experiments. So like because Borrelia is so specific, we tend to do very small experiments, but we're not answering all of the questions that we want to in one singular experiment. So we have to run a number of experiments. And so if we want to upscale these small experiments, the consistency may change. And that may change not only due to the changes in staff, but it can do with time and precision. And every experiment may have different results, even if you run it exactly the same. And optimization of like growth culturing or culture growth and media conditions is something that also has to be answered within these experiments. Um, and we think Opentrons can do that. So here is a video that I have going on of the Opentrons running. But as many of you are familiar, it's an open source modular, somewhat accessible liquid handling robot. And NASA can kind of explain a bit more as to how we were able to achieve our Opentron, or acquire our Opentron. But basically the solution that we think Opentrons present is that automated bacterial culturing and infection modeling can minimize human exposure. Therefore, our BSL-2 issue or having to train our scientists to be able to safely handle Borrelia can be solved with the robot taking care of most of that culturing. And Python programming makes it super accessible, or at least makes it easier to translate protocol language, scientific language into a code that is able to run these customized laboratory protocols. Um, and NASA, would you be able to kind of expand a bit on the process of getting these Opentrons?
Yeah, totally. I'll say briefly, like basically we purchased an Opentrons OT2 for the lab early on, and for the purpose of doing a bunch of optimizations of mostly protein expression assays. And then I guess probably like I don't know, maybe 2 years ago, we're talking to the Opentrons reps, and we're given the opportunity to upgrade to an Opentrons Flex because our machine was going out of service or something, and which we of course gladly accepted. And so now we have both the Flex and the OT2 that we can operate with.
Mhm. And both have been great for their applications, especially the Flex. So I have started like working on this project in the later times of the summer. And before this, I have my bachelor's of science in biochemistry and have only been trained in the wet lab space. And so learning how to program this Opentron as as a biochemistry trained technician had a big learning curve, let's just say. So in my undergrad training, I had taken only one Python course. And then I had online resources like the Opentrons Python Protocol API version 2 that I was able to reference. And so with my background knowledge of this one introduction Python course and this resource, plus the power of AI, in all honesty, I was able to figure out how I can make customizable protocols for bacterial culturing, in vitro transcription translation, PCR, and other microbiological experiments. So far, these are the two protocols or programs that I've been able to run on the Opentrons and have multiple experiments of and multiple trials with. And I'll be going over the first time we ran our media dilution set and media matrix protocol. So this was for E. coli culturing. So the media dilution protocol was first developed by an intern that we had in the lab over the summer, Sakti Subramanian. Sorry about that. And he first started the developing the general protocol of getting one solution of dye and diluting it down a row. And from his program that he wrote, I was able to expand it to do different variations. So in the 12 well reservoir here, we have different concentrations of dye, and each row correlates with each well. So we have 8 rows, 8 wells, and you can see that the dilution of that dye was successful for each row. And the Opentron was not contaminating as it went along and was diluting from one well to the other. But this was consistent with this standard 96 well plate. When it came to the 2 milliliter culturing plate, which is much larger, much thicker and taller, we didn't see the same we didn't see the same consistency. So moving from media dilution into creating this media matrix, um, which is just different conditions of the media that we were interested in looking at for E. coli, the conditions being sodium, magnesium, and glucose. Those were the three like sources that we wanted to vary in M9 media for E. coli. There, we see that there was growth on the left side of this plate, but then as we move forward, there isn't any growth. And although like the M9 media may not have been optimized for like maximum growth of E. coli, we still should have seen something because it wasn't toxic at the end of the day. And not seeing anything kind of correlate could mean that the dilution was just too was not accurate, or that the protocol itself wasn't being read as the same way that it would be on the standard 96 well plate.
Can I ask
And
you a question?
yeah, of course.
Uh, in that dilution with the E. coli, is the thing being diluted the media or the culture?
So the culture is being diluted. So
Okay.
every well here represents a different mixture of a different concentration of each component that we're talking about. So this is sodium, magnesium, and glucose. But we don't like the fact that we don't see any growth here means that we could have been missing like glucose altogether, or we could have been missing magnesium altogether. Um, and ultimately that ended up being what we saw. So we took the optic density measurements of every well before and after the 24-hour incubation. And we see that there's varied growth on the left side of the plate in the first 32 wells of the plate, which is expected. But then on the last couple rows and columns of the plate, it was roughly the same amount of growth. And we correlated this with the fact that the like as I was watching the protocol being carried out by the Opentron, the actual like pipette kept missing a well, or it would skip a row because the laboratory or the labware wasn't properly measured. Like it wasn't registered well with the robot. I took rough estimates with like a ruler and put that in as the measurements for that plate. But that wasn't enough. And so I had to reach out to the company to get the proper measurements. But I can expand on that a little more. So looking at the first 16 wells here, we wanted to test whether or not like, okay, these first 16, like during the run, they did run properly, and these were the wells that like worked best with the pipette. So we wanted to test that against this genome scale metabolic model that was available online, using the same variations and different concentrations of magnesium and sodium and glucose. We saw that the correlation of our experiment in the first 16 wells versus the genome scale metabolic model was about zero point 0.6. So roughly 60% of the time, we're getting what we expect out of these different concentrations of the components. So the best growth conditions, obviously for E. coli, was going to be for highly concentrations of magnesium and glucose. This is consistent with the literature. And yeah, we were able to replicate what the online simulations would say with the robotic model. But the errors and room of improvement was, again, the compatibility of labwares with the Opentron. We needed exact measurements to make this JSON file for the Opentrons. And so I had to reach out to the developers and to the companies of the particular labware that we had. So we wanted to be able to use what we already had. I didn't order any special labware just for the sense of cost. And I reached out to them. They gave me their entire like model, their diagram of the two mil plate. And I put in those exact measurements to see if it would work, if the pipette would operate better. And then sterility seemed to be good. Since there wasn't even any bacteria growth in the columns that weren't like idealized for E. coli, we knew that the antibiotic that we're using is good, so we'll continue to do that. And then sterilizing before with 70% ethanol before and after was essential to maintaining at least minimal contamination in these cultures. Bacterial distribution wasn't successful with the Opentrons pipette at first, like I explained. The actual pipette for the OT2 just kept missing and hitting the edges of the wells, and it didn't even aspirate the proper amount of media needed because the entire sorry, the word is slipping, but like the exact measurements needed weren't there in the Opentron. And so uh yeah, we I ended up having to replace the O-rings on the pipettes, which was a pretty tedious process. I also wiped down I had to remove the pipettes from the OT2, replace the O-rings, and make sure that any liquid that was inside, because the aspirating wasn't great. We weren't using filter tips either. Make sure any liquid was extracted from inside the pipettes, and then reprogram all of the labware once I got all the information from the companies that I reached out to. But that was a tedious process in of itself. And so like overall, was the timeline to process all this, I'll t touch about later. But then when we reran it again with all of these fixes, we started to see exactly what we expected, which was a successful growth of these 96 different conditions of 2 mil well culture. So to maintain sterility during its incubation, I covered the plate with this seal. It still had it's an air porous seal that allowed like for oxygen to be able to go through, but it prevented like any other thing from entering the media, which I thought was helpful. And we did see that there was good growth after a 24-hour incubation. So this is what the cells look like just out of the incubator. We do see varied size of pellets, which is what we expect depending on like the concentration of magnesium and glucose available in that M9 media. And then this is a version of the pelleted cultures, and we see overall that there's like pellets in every well, which is we're happy about. Like the media was properly diluted, the different media conditions were properly made, and the distribution of bacteria was good on this run.
Are
And it was
you
just
shaking? Do you have them in orbital shaker in the incubator?
yes,
Yeah, okay.
yes. We were shaking at we were shaking at 250 rotations per minute. We were using a very large 37-degree incubator,
Mm.
and and so that was our rotation speed. Yeah, for 24 hours. Um, and yeah, so like that's as I mentioned, this for the first run took a long time. It took roughly 3 months, mostly because I had to troubleshoot all of the labware that we had. Again, we were using the plates that were already available in our lab that we've bought in the past for different experiments and or that have been donated to us from other labs. Um, so that took a while to get all of that information from the companies and like look up the dimensions and the exact measurements of every plate. Network connectivity and IT coordination was also a big hurdle since we were moving the Opentron from our upstairs shared lab space into a microscope room that was on the first floor of the steam plant. We had to reconnect the Opentron to the network in order to communicate with it from my laptop. And so that took a long time, and IT was very confused as to what the Opentrons was and its purpose, and they wanted justifications for everything. So that took roughly a week, and then that after I was able to get it connected during that time where I was trying to get it connected, I was developing the protocol in of itself and writing up the code and learning how to use the Opentrons Python API. And there is online simulations that you can do. So Opentrons does provide a program that you can run in your command line where you can simulate a run from based off that protocol. You still have to upload all of the labware information into the same folder as where you have your protocol so that the simulation can read everything all at once, like how the Opentron will. And that took a while to like get to work, but while IT was struggling, I was able to run all of those. And then uploading the protocol onto the Opentron and getting to properly read and work took a while. And setting up on the app also took a while in my laptop. And so it took roughly 4 weeks to just get all the technical stuff done. And then after that, it was actually doing the physical runs and troubleshooting all of those. So this took roughly 3 months to be able to get. But once I had that first run, I then was able to share the results at a conference, and people were very interested. And then once I came back from the conference, knowing having everything, the whole Opentron system set up and having the infrastructure already ready to go, I was able to do a second attempt in less than a week. So I pulled those same protocols that I already had from my repository, then ran test runs in the simulation and has some data validation from the simulation. uploaded that onto the Opentron, and then ran all the protocols within the same day. So like ideally, this wouldn't even take a whole week. Like you could do all this within a day, but I was running other things at the same time. So this was kind of like a side project that I was able to do on top of my regular responsibilities. Um, yeah. So like once the system was already set, the protocol development workflow was pretty straightforward from here. The first thing is to check the repository, which is our Notion page, and then we also have a shared file that people can access through the Rhino computer on our laptops. But essentially, check the repository if a protocol exists for what you want to do. If it does exist, great. Just upload it onto your Opentrons app and then run it. If it doesn't exist, ideally, if it doesn't exist, like you don't have the labware or it's not customizable, you can then edit that protocol that like the skeleton of that protocol in Jupyter Notebook, and then using Opentrons Python API as well as AI, but you have to train the AI to kind of understand what the Python API is. To then like adjust it to your needs. If it doesn't exist, but it's available online, for example, it exists on the Opentrons protocol library. Um, it may not be customizable if you just straight up download it and then run it on the app. So you want to be able to download the program into your local machine and then edit it in Jupyter Notebooks in the way that you need, upload that to the Opentrons API, and then go to the robot. And if it doesn't exist altogether, that's when you would start a new project, like from your Jupyter Notebook, and then go straight into customizing like labware, making sure that like the labware that you do have, it can be compatible with the protocol that you want to run. Get the tips and the consumables, and then test your new program in a simulator before uploading it onto the Opentrons API and then running it on the robot. Ideally, like we're at this stage right now where we have protocols that exist or are I'm able to like use the skeleton off the Opentrons protocol library, but it needs to be customized. And so downloading those programs and changing, like tweaking it a little bit so it's able to adapt to what we have in our lab, it becomes a much faster route once like the Opentrons has already been set up and the API is already working in live. But once you're able to execute your protocol on the Opentrons, from there we're able to like run our samples in the way that we need. So that could be through LCMS, that can be through Flow, that can be if we're doing protein expression, protein gels, ideally, which is what I'm working on now, and then analyzing that data from there. But that's just the robotic automation part. Our next goal is a machine learning integration. So like what do we do with the data once we already received all the information that we wanted to out of our experiments? Essentially, we want to build this online learning in vitro experiment model, aka Olive, that a system that iteratively builds a series of compressed sensing data that we've made, and collecting and reconstructing that data using fewer measurements than normally needed. So every Opentron run would give data that this algorithm would take in and be able to use that data to build a better experiment, and then run that through the Opentrons and do that iteratively and every experimental run until we have an experiment that answers our specific question. This goal introduces new learning curves. So we want an algorithm that can do quality check, store, and learn from a large and diverse set of experimental data. So as I mentioned, we wanted to be able to do LCMS and then as well as protein gels and flow cytometry with our samples. And that's a lot of data to be able to process. And so this algorithm has to have the capability to do that really quickly. We also want to be able to integrate it with Opentron specific Python language. So if it's using one language, it should still be able to translate into the Opentron specific language so that there's less effort on our end to try to translate what our algorithm is saying and give it to the robot. This experimental loop, we want it to be able to improve on each iteration and improve on itself, so that given it's a scientific question, it'll be able to design an experiment or at least go through the process of designing a better experiment every time that we run something. But yeah, these are all learning curves that I still have to meet. And like I said, like it took a long time to even get the Opentrons running. But now that it is running and we're able to integrate new programs, this is the next step that we want to be able to do with the data that we have. But yeah, so we have a system that is small scaled but multifunctional. The Opentron, we've been able to adjust it for bacterial growth, but now we want to do protein expression. We also want to include that gas exchange control, so the 3D printed manifold and custom labware, which is what NASA has been working on printing on some of these, but essentially cutting the cost. We don't want to rely too much on like using an Opentrons as a direct source for all the labware that we want. We still want to make this accessible, make this model accessible to community labs such as like Sound Bio in Seattle. But like these 3D printed manifolds that have been used in previous publications seems like we want to be able to integrate that, and seems like a promising method that we can use. The learning algorithm to build new protocols is our next step. And then integrating real-time data measurements like 24-hour OD measurements would be great to determine how well bacteria is growing in like within 3 hours or within 5 hours of its growth period, as well as pH colorimetrics. So the Flex does have a camera attached to it, and we just got that up and running. And so if we're able to use like a pH colorimetric system in real time, we'd be able to determine like the quality of that experiment based on the pH that we're getting. But yeah. I just wanted to shout out my lab and everyone in it. This project wouldn't have been able to like proceed and grow in the way that it has without the support of my lab members. And then also shout out to Sakti, who had essentially ignited the fire for this. And yeah, thank you. And here are my references. Open any questions.
Oh, good documentation. All right, let's thank Angie. Thank you very much. Get some reacts, some claps. All right, I will stop the recording and then we can have questions and then we have a little bit of time to talk about some grant opportunities also.